Hello to all your students. So in this video we'll be discussing about the mechanism of action of those antibiotics that mainly acts by inhibiting the bacterial cell wall synthesis. Now we have gram positive and gram negative bacteria, and like most of the bacteria falls in these two categories. Now this outer layer of the bacteria is named as cytoplasmic membrane, and there is one more covering beyond this cytoplasmic membrane which is named as cell wall. Now as you can see the differences in the cell wall of gram positive and gram negative bacteria. In gram positive bacteria the cell wall is thick and porous, whereas in case of gram negative it has a thin cell wall. But the this gram negative bacteria have got an additional covering, that is the outer membrane, which is lipid in nature. Now this makes the difference. Gram positive bacteria have thicker but porous cell wall, and gram negative bacteria has thinner cell wall but has an additional lipid covering. And as you people might be knowing that the nature of lipid covering is, if any substance wants to cross this lipid layer, it should be lipophilic in nature or it should have very smaller size, right? Now suppose you have consumed any kind of antibiotics, and if the organism is a gram positive bacteria, then this antibiotic it can easily cross across this porous cell wall and attack the bacteria. But if the organism is gram negative bacteria, then most of the antibiotics it cannot cross the lipid outer membrane. But this does not mean that the gram negative bacteria are protected from antibiotic attack. God have made doors that is present on the surface of these gram negative bacteria, and these doors are named as porins. Okay, now we shall discuss about these porins any other time. Another important structure is the space between the cytoplasmic membrane and the cell wall, which is named as periplasmic space. Next, we shall now have a little detailing about the cell wall before we move forward. So taking a gram positive bacteria, this cell wall is basically composed of a backbone which is made up of sugar, and sugars are also known as glycan. Now this sugar backbone are interconnected by a bridge which is our peptide chain. So together this bacterial cell wall can also be called as peptidoglycan cell wall, since it is composed of glycan, that is a sugar, and peptide chain. Next, you people might be wondering that why we are giving so much importance to this bacterial cell wall, why this bacterial cell wall is so much important. So the answer to this is bacterial peptidoglycan cell wall protects them from bursting due to osmotic pressure. Like suppose this is a gram positive bacteria, and when an antibiotic approaches it, it will start damaging the peptidoglycan cell wall. Okay, it will go on damaging it, and it will finally approach to the gram positive bacteria. So there is no cell wall present here. Okay, when the entire cell wall is inhibited and the bacteria interacts with the environment directly, so there will be movement of fluid inside this organism, and because of osmosis and osmotic pressure increases, and due to which this bacteria will swell and eventually this bacteria will burst out. So therefore this cell wall is very much important to keep the bacteria alive. This cell wall will protect the bacterium from the osmotic pressure burst. But why should we keep it alive? We want to kill these disease causing bacteria, right? So one way of killing these bacteria is by inhibiting their cell wall. Now first of all we shall check out how the bacteria synthesizes the cell wall, like what all steps are involved in the bacterial cell wall synthesis, and then later on we will find how various antibiotics will inhibit the bacterial cell wall synthesis. See, we have got different antibiotics that will interfere with the different steps of the cell wall synthesis. So first of all, we shall check out how these bacteria are synthesizing their cell walls. So now imagine this is a gram positive bacteria. There is a monosaccharide which I have shown in green box. Okay, so from this monosaccharides it synthesizes a very important constituent which is NAM and NAG. NAM means N-acetyl muramic acid and NAG means N-acetyl glucosamine. Next we need to activate these two sugars, and for activating it we are just attaching a UDP group to it. In addition to this, we have got a peptide chain which consists of 5 amino acid. So this peptide chain of 5 amino acid is been attached to the NAM moiety. Okay, so what we did so far, we started from a monosaccharide, and from that monosaccharide we synthesized two important sugars, that is NAM sugar and NAG sugar. NAM means N-acetyl muramic acid and NAG means N-acetyl glucosamine. And next what we did is we activated it with the attachment of UDP group plus NAM sugar. To it we have attached a peptide chain which consists of 5 amino acids. Next thing is this bacterial wall, this bacterial cytoplasmic membrane have got an important protein, and the name of this protein is bactoprenol phosphate. Now the main role of this protein is to attach this NAM and NAG sugar. Okay, so this NAM and NAG will be attached, UDP will be removed out, and this NAM NAG attachment so formed thing is known as the peptidoglycan precursor. Okay, so the main role of this protein is to synthesize peptidoglycan precursor. Now its work have not ended still. Its another job is to transport this precursor moiety. Okay, so the main role of this protein is it have got two works to do. First one is to synthesize this peptidoglycan precursor, and this synthesized peptidoglycan precursor it has to transport it outside, that is in the periplasmic space. Okay, so in this manner it will be synthesizing n number of peptidoglycan precursor. Now next thing, next step in the synthesis of this bacterial cell wall is this yellow star. This yellow star is nothing but it is a kind of enzyme which will be acting at this part. Okay, at this part. So what basically it does, it will glycate, it will stick, it will attach the two peptidoglycan precursors, two moieties it will be attaching. So this enzyme it will come, it will act over here and it will attach this peptidoglycan precursor. Okay, it will go on attaching these peptidoglycan precursor. Okay, so this enzyme what it is doing, it is producing a glycosylation reaction. So we can say it as glycosylase or transglycosylase. Okay, so this is the enzyme what it is doing, it is attaching the sugar backbone, it is making the sugar backbone. Now next thing is performed by this pink star. Okay, so this pink star is also a kind of enzyme. Now what this enzyme is doing means it is acting at this area. Okay, so what basically it does is it will act on this peptide chain which is composed of 5 amino acid. Okay, and meanwhile what it will do, it will remove the terminal amino acid. Okay, so on the left side you can see these 5 peptide, this peptide chain consisting of 5 amino acid. So basically the fifth amino acid, that is D-alanine, it is been removed out. This terminal amino acid is been removed out, and after removal the both the chains will get ligated. Okay, so as you can see each a peptidoglycan precursor molecule have got a peptide chain which consists of 5 amino acids. So what this pink star enzyme what it did, it removed one amino acid and it linked the chain. Okay, so this so formed linkage is known as peptide cross linkage. Okay, so the work of this enzyme is to remove a terminal amino acid and to produce a peptide cross linkage. So the name of this enzyme is transpeptidase. This enzyme have got one more name, that is penicillin binding protein. Okay, so basically what this enzyme does, it will produce a cross linkages between the sugar backbone. Okay, whatever sugar backbone is being made, it will connect them together. Okay, so that is transpeptidase reaction, and this enzyme, it is also known as penicillin binding protein. Okay, and this is how the cell wall is been synthesized in the bacterium. Okay, first of all, NAC has been produced, and then they are been attached, means peptidoglycan precursor has been formed, and then they are moved outside the bacterial cell, means in the periplasmic space, and then these precursor molecules are interconnected with the help of transglycosylase enzyme and to form a sugar backbone. And after formation of sugar backbone, peptide cross linkage will be taking place, and in this manner we are synthesizing the bacterial cell wall. Now we shall check out various antibiotics that inhibits the bacterial cell walls. So there is a list of antibiotics that will produce a bacterial cell wall synthesis inhibition. So these antibiotics are beta lactams, bacitracin, cycloserine and vancomycin. Now we have got a mnemonic by which you can remember all these four antibiotics. They are bind to bacterial cell wall. Okay, so beta lactams, bacitracin, cycloserine and vancomycin. Now we shall start our discussion with the first antibiotic, that is beta lactams. Now the two important beta lactam antibiotics are penicillin and cephalosporin, and we know that these two antibiotics, they mainly act by binding with the penicillin binding protein, and which is nothing but your transpeptidase enzyme. So basically what these two antibiotics will do, they will inhibit this transpeptidase enzyme, and just now we saw the main role of this enzyme is to create a peptide cross linkage. Now when we gave a beta lactam antibiotic, so what will be the scenario? There won't be cross linkage, means there is no cross linkage, no strong cell wall, and therefore bacteria can undergo osmotic burst. Okay, so this is the mechanism of action of beta lactams. Beta lactams like penicillin and cephalosporin, they mainly bind to the penicillin binding protein, and which is nothing but your transpeptidase enzyme, and they will inhibit it will stop the transpeptidase enzyme to do its work. So this is the mechanism of action of beta lactams. Now our next antibiotic is bacitracin. So let us check out the action of bacitracin, the mechanism of action of bacitracin. So the main action of bacitracin, before knowing that, we should know a small thing about bacitracin is mainly used locally. They are used only locally. They are never used systemically. Sorry. Now this bacitracin, they mainly act by chaining or locking up this protein bactoprenol phosphate. Okay, so it is inhibiting this bactoprenol phosphate protein, and we know what is the main role of bactoprenol phosphate protein. Yes, their main role is to synthesize the peptidoglycan precursor and to transport them outside. This is the main role of this protein, but this bacitracin is inhibiting this protein to do its work, and therefore because of which the thing what the bacitracin will create is no peptidoglycan precursor formation, no transportation of the precursor in the periplasmic space, and therefore the cell wall synthesis will be inhibited and bacteria will be prone to the osmotic pressure burst. So this is the mechanism of action of bacitracin. They are mainly inhibiting this protein bactoprenol phosphate. So this was all about the second antibiotic mechanism of action. Now coming to the third antibiotic, which is cyclosporin. Now this cycloserine, it is mainly used as a second line anti-TB drug. It mainly acts over here. Okay, we know this as a peptide chain which is made up of 5 amino acid, and each amino acid is made up of different enzyme. So cycloserine, what it does is it mainly acts by inhibiting the enzyme which is required for the synthesis of D-alanine. So there won't be any D-alanine in this peptide chain. So here also we do not expect this D-alanine, means there won't be 5 amino acid, just 3 amino acid peptide chain will be there, and therefore we can say that what cycloserine will do, it will produce an improper peptide linkage, and cross linkage is not possible. Therefore we can say cycloserine causes all this scenario, and thereby no cell wall synthesis will be taking place, and bacteria will be prone to the osmotic pressure lysis. So this was all about the mechanism of action of cycloserine. Now coming to our last drug, that is vancomycin. So vancomycin, it mainly acts over here in this place, and what it does over here is it prevents removal of terminal alanine amino acid. It will come and it will bind with the d-alanine, and it will inhibit its removal, which was been taking place during transpeptidation reaction. Plus it also works over here, means it will also inhibit this enzyme which is required for glycosylation reaction. Now first of all we shall check out the first area where it prevents the removal of the terminal amino acid. Now we know that this enzyme, it is mainly required for cross linkage, and how it is producing cross linkage. Peptide cross linkage means it will act over here, it will cause a removal of the terminal amino acid, and thereby linkage will be taking place, isn't it? So this was peptide cross linkage, it was taking place. But what this vancomycin will be doing, it is inhibiting the removal of terminal amino acid, and because of which there won't be any cross linkage possible. Now next, another thing is it is also inhibiting this enzyme which was required for glycosylation reaction, isn't it? So we know that what this enzyme was doing, it was binding this peptidoglycan moiety, okay, it was producing the backbone. But what this vancomycin is doing, it is inhibiting this backbone formation, it is preventing the stitching of the peptidoglycan precursor. So for vancomycin we can say that no cross linkage possible and no glycosylation will be taking place, and therefore there won't be any cell wall synthesis, and bacteria are more prone to undergo osmotic pressure burst. So this was all about the mechanism of action of all these antibiotics. I hope you people found this video helpful. Thank you for watching.